One more.
We have started the live session as well. So if you miss the meeting or you get disconnected, then you can join the meeting via the YouTube link as well. Thank you. We would be starting in just 2 more minutes. I welcome you all to this free webinar on machine learning for biomedical drug discovery. This innovative summer online program that discusses several key technologies relevant to modern biomedical data science research is designed to modern biomedical data science to introduce participants to the application of data science to important challenges in clinical and pharmaceutical applications using practical examples that cover genomic, transcriptomic, and structural data. This collaborative program has brought to you by Pine Biotech USA, FABA, Academy India, Pharmatoppers India, and Probiotechnica India. We now live in a world that is recording data every day. The healthcare data related to the clinical trials, electronic medical records, EMRs, genetic information, care management databases, billing, internet research, and social media data is growing massively and pose a challenge to the community of students, scientists, and researchers. This challenge is also an opportunity to learn and discover from the vast amount of information that is now available publicly. After working for last several months with a group of experts and industry leaders, we are now excited to bring this unique opportunity for students to learn and work on real data discovery pipeline for a disease or an indication. To give a brief introduction of the associations, Pine Biotech is a US-based company that provides resources, solutions, and training for research and education in biomedical informatics. We have been developing an innovative approach to incorporate bioinformatics training, educational materials, and biomedical specializations for universities, companies, and top research labs. FABA Academy is a special wing under the FABA organization that focuses on grooming the students of the universities nationwide and make them future ready. FABA Academy prepares the students by providing skill development and professional development programs, which gives them the confidence to rule the world of pharma and biotech industries. Pharma Toppers, a pioneering institute committed to provide refined education to pharmacy aspirants and training programs in bioinformatics, pharmacovigilance, etc. Probiotechnica Research Private Limited has a mission to equip and empower every student and faculty with the latest technology and tools to create an edge from others. The company strongly believes in serving society with quality work so that the industry can get the best out of its resources and to make students industry ready. Schrodinger's physical-based computational platform integrates differentiated solutions for predictive modeling, data analytics, and collaboration to enable rapid exploration of chemical space. The platform powers our own drug discovery efforts from target identification to hit discovery to lead optimization. It also drives our research collaborations to develop novel medicines for critical public health needs. Now, let me introduce you to our scientist from Schrödinger, Dr. Prajwal Dandekar. Dr. Prajwal Dandekar received his master's and PhD from the National Institute of Pharmaceutical Education and Research, Naipur, Mohali, in 2010 and 2015, respectively. He is a recipient of the prestigious DAD Fellowship to perform research at Heidelberg University. Dr. Pritesh Bhatt, he is currently working as a senior scientist at Schrödinger, Bangalore. His doctoral research involves the identification of inhibitors for various enzyme targets of Mycobacterium using different computational modeling approaches at Birla Institute of Technology and Science, BITS Pilani. Mr. Vinod Devraji, he's a senior scientist at Schrödinger working with computer-aided drug discovery research for 9 years. He completed his M.Pharma with Madras Medical College of Pharmacy. He has research experience in topics such as computer-aided drug designing, DNA intercalations, organometallic compounds, and biologics. Personally, he is holding 30 research publications in reputed international publication groups to his credit. Dr. Kaushik Kasavachla is currently working as a senior scientist I at Schrödinger at Bangalore. He received his PhD in chemistry in 2020 from Stony Brook University, New York, USA. He holds 8 publications to date, with 2 more in preparation. His work has been cited more than 3,500 times. His research interests include developing enhanced sampling methods and force fields for biomolecular simulations, and designing antibodies and drug molecules. Mr. Kishore V. Associates Solutions Architect provides technical support to users. The program will leverage our top-rated online tutorials using the Bioinfo portal with smart tracking of activity and curated datasets designed for a great learning experience. In this program, we will combine online sessions, self-guided study materials, and practical assignments for an immersive experience that has been proven to be effective in our well-known and respected Omixlogic programs. We will learn important concepts within biomedical research, infectious diseases, cancer research, and learn about the immunology of infection, molecular disease markers and stages, and how these diseases can be diagnosed and treated using machine learning approaches. For different parts of the sessions, we will leverage Omixlogic resources that you can use to learn about machine learning, asynchronous coursework with theoretical material, quizzes, and practical assignments, our cloud-based analytical server where you can analyze large genomic data and visualize results, the Omixlogic code playground where you can practice coding in R and Python, as well as repository of project example. So let's start by signing up to learn.omixlogic. This educational portal will help you get started with your bioinformatics journey. Now I request you all to please go to the server omixlogic.com. You can type in omixlogic.com. Now, those who are already signed up into this server, they may sit back, and those who are not signed up, I request them to please sign up yourself in this learn.omixlogic.com portal. Once you are done, please send a yes in the chat box so that we can proceed further. Thank you so much, Dr. Harpreet. Dr. Harpreet has shared the link of the portal learn.omixlogic. Please sign up so that we know that you are signed up. Please send a yes. I can see a lot many responses. Let's wait for 2 more minutes for others to sign up as well. Signing up on this portal would help you get the access of all the courses and also let you know about our upcoming programs and trainings, and you can easily register yourself here for the courses or the trainings. Those who have signed up and those who have not can take 2 more minutes to do so. Yes, I'll shortly share the Slack channel link so that you can join. For others, we also have a Slack channel where we communicate directly and tell about all our upcoming courses and trainings. I will share the link soon. So throughout the course, you will be given the directions to access the TBioinfo platform for bioinformatics processing and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections to this multi-omics platform. And in each course, you will load, transform, and visualize data in R and in Python. Now, I would like to invite our CEO, Ilya Brodsky, to discuss more about the program on machine learning for biomedical drug discovery and also share the details of the sessions. Over to you, Ilya. Thank you so much.
Thank you very much, Monica. And welcome everybody to this webinar. So I will just kind of briefly introduce some of the topics to give you a background on what we will be offering during the data science program starting on June 14th. And to briefly introduce you on my part of this presentation, I want to give you a little bit of a background about Pine Biotech, what we do, and how did we arrive at the Omixlogic portal idea, so that you will be oriented in the program itself and think about some of the applications of data science and biomedical research. So Pine Biotech is a technology transfer company. Originally, the company was formed to commercialize research coming out of the Tauber Bioinformatics Research Center at the University of Haifa. And our team has been working for about a couple of years to make sure that the same tools that are used in many cutting-edge research projects in biomedical data analysis are being available and come with training for students, researchers, and clinicians. And so the program that we developed has been tested and piloted in many different settings, including the Louisiana Biomedical Research Network, different programs like Georgetown University Systems Medicine, as well as universities around the world. And so as a result, we have received a lot of feedback that we've incorporated into the way that the training program has been structured. But our company doesn't just focus on training itself. And so as a part of various different research projects and collaborations, we have been privileged to work on a number of research application development for different areas that I will be actually talking about in just a few minutes. One of these has to do with the integration of omics data in a variety of big data research projects. As you can see here, this is integration of different types of data, phenotypic data and data about omics analysis. We've worked on a number of collaborations for biomarker discovery in real-world evidence collection and clinical trial projects, as well as projects for small molecule screening and this hit-to-lead optimization from libraries of small molecules. And so as a result of this kind of research experience, we really developed this kind of a data science program that is intended to give an opportunity for students that come from high school, undergraduate, as well as postgraduate level, teachers and faculty at high schools, colleges, and universities, as well as researchers like biologists and clinicians at research labs and biotechnology companies, an opportunity to master tools and skills that are required for effective data-driven research projects. And so to speak a little bit more about the way that this program is organized, I want to give you a background on what Omixlogic as a training portal really is. So now that you've all signed up to the Omixlogic portal, let me introduce what the programs, courses, and different training resources that we have, how are they organized, and what's the difference between them. So the goal of Omixlogic programs is to provide training in bioinformatics, and I'll talk a little bit about the relationship between bioinformatics and data science in a little bit. And the important part of it is to enable independent research guided by mentors and peer examples. So it's more than just a collection of courses, it's also a community. And this community has a shared passion for data-driven research and an appreciation of citizen science. And so what's important here is that this is a community where you can get to know other people, you can collaborate with other people on research projects, and a critical part of that is essentially training. And one of the challenges that we see is that many people from different backgrounds have to apply different types of data science skills to develop independent research projects using latest technology in life and data sciences. And so one of the programs that we've developed as a result of this kind of vision for this Omixlogic community is a program on Omixlogic data science. And this kind of a program combines resources online, asynchronously, and in a blended fashion to empower different types of people with different backgrounds to learn about statistical analysis and machine learning, experimental design and technical variation that is to found in exploratory data analysis, as well as to apply data mining tools for finding meaningful patterns and associations, and study complex biological systems using big data. So this program on data science combines a number of different important aspects, including different types of omics data, steps for exploratory data analysis and processing, data analysis for big data integration and mining, and the application of these different tools to different types of research questions. So today's webinar is going to focus on a number of these aspects, which we will do an introduction to data science and bioinformatics. So what's the relationship between data science and bioinformatics? A little overview of data processing, cleaning, and exploration, how to do that, and what are some of the tools that we will cover in this program. The application of data science in biomedicine, introduction to machine learning algorithms for next generation sequencing data, the application of data mining and integration for multi-omics or X-omics integration. And then I will pass it on to our next speaker to talk about deep learning, natural language processing, and image analysis, and how those are important in this domain of biomedical data analysis. So let me first start by speaking about the relationship between data science and bioinformatics. So what is data science really? Data science is data storage, organization, and retrieval, including application of different data formats, biological specialization, right? If we talk about the domain where we apply data science in the context of biomedical data science, right? This includes biological research, but it could include many other different domains, right? And so here, what we all can appreciate is the similarity between data science and bioinformatics, because all the same different aspects of data science, if they are exclusively applied to biological data, now we can call that bioinformatics. And so when you think about bioinformatics, really that talks about different types of information technologies applied to biological or biomedical research. And so one of the differences where we see the importance of these kinds of skills in information management, in statistical analysis and machine learning is the specific application of these methods to major impactful questions in biomedical data. And so two main areas that I want to focus on today are drug discovery and clinical trials, because I think that in both of these areas, there is a major change or a paradigm shift that is going on, and many of you are probably aware of these transformations, but they really require this change in thinking about the way data is used for these two problems. So what is the challenge in drug discovery? One of the major challenges in drug discovery is the time. How much time does it take to get from a number of potentially active compounds to a compound that could actually be brought to market through the different stages of commercialization of that compound? And that goes through stages of research and discovery, where multiple compounds are analyzed and several are selected, to then hit to lead optimization, and then through several stages of clinical trials. And so the clinical trials area is the second major area where this transformation is quite obvious. In clinical trials, you have to design a specific process by which that compound that is not toxic and is efficacious can go through several stages of validation, and that requires selection of the right types of candidate patients, the right kinds of indicators, and the right kind of validation for a certain level of efficacy for those to proceed from stage 1 to stage 3, and then to be observed in post-market surveillance to understand how other types of patients are responding to this type of treatment. So these two areas are very important in the area of data science for biomedical data, and those are some of the things that we will focus on in this program. Now, importantly, I think there's a huge opportunity in regards to this kind of big data, because a lot of this type of data is actually available on publicly available repositories. And not only is it available, we know that since this pandemic, the amount of data is actually growing. And so this accelerated pace of availability of big data that is now accessible to anyone who is interested in asking research questions about this type of data provides us also with an opportunity to develop these kinds of training programs where instead of giving you test examples, we can actually leverage some of these real-life project examples from the research and clinical areas to provide you with an immersive experience where you can apply specific types of data science tools and methods to ask questions about these kinds of data sets. So the first challenge, having this kind of availability of data, the first challenge that I see is the challenge of data structures, how to clean, process, and normalize the huge variety of data types that is available. And these include imaging data, streaming data like EMR, clinical data, omics data, chemical data, phenotypic data, all have to be prepared so that they are structured and available for the layer of analytics that we would like to apply in a meaningful way to these types of data sets and potentially even integrate them to find associations between the phenotypic data and the different types of omics data that are available. And so one of the things that our company has been focused on is making the omics data simplified, democratized, and more clearly understood by the millions of researchers that are out there trying to understand the molecular drivers of disease, as well as molecular indicators of treatment efficacy. And so the first challenge in omics data that I see is the variability of data types and what do they actually represent. So since they provide a very detailed view of molecular changes and activities inside the cell, we're talking at a subcellular precision that comes with a lot of challenges. And so what are these omics data types? One type of omics is phenomics, where we can look at cell morphology and characterize different cell shapes and activities. We can look at genomic data, which is essentially variation at the DNA code. We can look at epigenomic data, which essentially tells us about subgenomic regulation, so regulation that is not inside the DNA code itself, but provides a layer of modification like data sets like whole genome bisulfite sequencing and chromatin immunoprecipitation, where we can understand different types of variations. Transcriptomic data, the result of expression of mRNA and downstream modification of that mRNA. And so as you can see, these data sets are very different. They're different because they describe these different aspects of the same biological phenomena, but using different technologies. And so the first part of this program, we will do a little overview of how to process and structure such data so that it is ready for integration. And so here we will look at several different examples of genomic data like whole exome sequencing and whole genome sequencing, epigenomic data like DNA methylation and ChIP-seq, transcriptomic data analysis for single cell and bulk RNA sequencing, as well as proteomic and metabolomic data. So all of them coming from the biological material and being processed into a structured data set. And so now once the data set is structured, now we enter the domain of data science. And in this domain of data science, now we can operate with the same statistical data exploration and classification methods that allow us to ask questions about relationships of these different and distinct data types that describe different types of biological phenomena, but also different types of biomedical applications, about recruitment of patients for clinical trials, about what drives disease progression, and about what kinds of treatment targets are there available to be able to intervene in a specific malignant process. And so as an example, we can take a look at how this is changing the way we can treat patients. Ultimately, when we generate new data, instead of coming as a new set of data, we essentially see that as an integrated data set combined with other existing samples that allow us to ask questions about population, similar patients, patients that have benefited from different types of treatments, and the features that define those patients, or different types of markers that we could rely on to optimize future treatment. So Omixlogic is essentially a complete roadmap for data science, an application to biomedical research data that will talk about finding data sets, exploring and visualizing those data sets, understanding different types of research methodologies, and gain insight and understanding, looking at some of those questions that are being asked by top medical journals and research studies that we can find in the public domain. So let's take a look a little bit at what the Omixlogic portal will show you as you consider joining this upcoming program. So once you have registered on this portal, the Omixlogic portal, you will see here a number of different resources that we will leverage in this upcoming program. And this includes different research project examples, processing tools for big data analysis that you will gain access to throughout our server portal, which is a server for big data analysis, different coding exercises that will allow you to take process data, perform data exploration, data visualization, and analysis, and looking at different kind of case studies that show how these different methods are applied to different types of research questions so that we can answer them in a data-driven way. So first of all, there are many different applications, including oncology, infectious diseases, neuroscience, and agriculture. And in each one of these categories, we will take examples of projects where you can understand the specifics of the data that is being generated and the specific questions that are being asked. The first challenge that we will overcome is the challenge of big data. And there we will utilize the user-friendly and logical user pipeline builder that will allow us to do sequence alignment, analysis of gene expression data, or even analysis of the microbiome to structure that data and prepare it for analysis. Then we will proceed to look at different types of coding languages like Python and R to understand how to load that data, explore, and analyze that data in a meaningful way. So let me give you an example of what that looks like. If you go right here to the Courses tab, you will find here a number of examples for what I am describing here. So you can see different types of projects that we will give you access to, and you will see some examples of the coursework that you can take a look at when you subscribe to the program. So let's take a look at some of these examples by looking at this course called Introduction to Data Science in R. And I will paste this link in the chat. Okay, so here, once you have reached this page, you will see some examples of different lessons that we will go through, including how to load data and check for different types of variables, how to visualize data using basic and more advanced methods, how to explore data using tools like principal component analysis and visualization, different statistical relationships, how to perform kind of specific methods like differential gene expression, clustering, k-means and hierarchical clustering, some examples of unsupervised machine learning, how to perform classification with random forest and other types of methods, and all the way to an understanding of how different statistical tests can help us extract meaningful features from our data and interpret them in a biological way. So you will find these different examples. These are courses that are free, that you can try for free even before you join the program. And then some of the more advanced methods that are highlighted here with a star, you can use once you either get an asynchronous subscription, which is very affordable, or proceeding to look at some of our programs. So the programs that we offer, this is an example of a program where you will be guided by mentors that are experienced in a particular domain and explain specifics of how this data is applied to different examples. So the benefit of joining a program is not only gaining access to the different resources that you are seeing here on the screen, but also being able to interact with the mentor and the participants of the full program. And once you interact with them, to be able to schedule a session with that mentor, to have one-on-one mentoring with that person, also being able to interact with other participants of the program by joining the Slack channel. And so here you will see a program overview. So this is a link to the specific program that we are starting on June 14th. So you can see right here, starting on June 14th. You can see a little overview of this program right here in the video. Here are the different sessions that Mohit will describe in just a few minutes. And then as you scroll down, you see some of the access that will be provided as a part of this program training. So you will combine asynchronous materials and in-person sessions that are described there. Now, many of you are probably wondering what are the different options and how to complete your registration. And so for that, I want to make sure that we can answer all of the questions in the next maybe 30 or 45 minutes of this webinar and explain anything that you will need to know to gain the most benefit out of this program. But before we do, and before I pass on my part of the session to the next speaker, so Nalika, if you could start the poll so that we can get to know some of the participants in this webinar today.
Yes, sure, Ilya. Thank you so much. So I've just started the poll. I've got 3 questions for you all, and I would want you to answer these so we can know you better. The first question is, what is your educational background? Graduation, post-graduation, PhD or postdoc? And my second question is, what are your research interests? And the third one is, what is the reason you are interested to attend the training? This poll would be on for more 2 minutes, and I request you all to please answer the questions. Thank you.
Okay, thank you very much. Now, all of you that have already answered this question or this poll, I want to invite many of you that are interested in learning some of these tools. Many of you are interested in many different aspects of the program, including learning how to code, gaining advanced understanding of machine learning and deep learning, receive a certificate, mentorship, and all of the above. So I see that the majority right now, all of the above. Okay, and so before I answer some of the questions in the chat, which I will do in just a few minutes, for those of you that have completed the poll, I want to connect with all of you in a professional way on social media. And so what I would like to ask is that all of you join our group on LinkedIn, which is a group where you can find a lot of the mentors that we have, as well as a lot of the participants of previous programs. So if you could please join this group called Omixlogic. That will allow you to see some of the information that we will have about upcoming sessions, opportunities to join different types of research fellowships and internships that are available over the summer, and also interact with professionals that will be mentoring this program, and potentially you could benefit from getting to know in the course of the next program. So a great resource that is beyond the training, obviously free. And if I could ask Sonalika that you also post the link to the Facebook group as well. Okay, so let me now go through some of the questions that are in the chat and make sure that I answer those as well. So I see a lot of questions here about certification and different types of courses. So I will leave that for the later part of today's webinar. Dr. Mohit Mazumdar at the end of the webinar will explain some of those things and how to join, how to register, what are the different sessions, and how to get certification. You will get the certificate, and this certificate will be partly automated through our system where we track all of the activity, and partly will be also approved by the mentors that will be guiding this program. For those that are coming from computer science background, this is a very interesting question, so I want to just pause right there. So it's very important that many of these methods in data science are widely accessible through many different tutorials and many different systems online. And so that is perfectly fine if you're already a data scientist or an engineer. The question becomes, how do you apply these skills effectively to an area of biomedical discovery, including the applications to pharmaceutical and biotechnology companies, or clinical diagnostics and clinical research? And so the main answer to that question is that this whole curriculum is very project driven. So what we will do is we will introduce you to the different data types, but also experimental design and the questions that are being asked in the research papers that we leverage, so that you can actually apply the methods that you probably already know, but to the different data types and the different questions that are being asked, maybe in a domain that you're not so familiar with already. So VM Dev, hopefully this answers your question right there. Sue Barsh is asking if I could explain normalization in a few sentences. So definitely normalization is a critical part of preparing your data for analysis, because many times when you deal with different laboratories, different types of settings, data that is being generated in the different settings is affected by many technical variables that we don't have any control over. So if you think about distance, if I measure distance in the US, in India, or in China, it's going to be the exact same distance. But if I measure gene expression, that gene expression is going to be subject to the technical things that I have in my lab, the way I collected the biological sample, the timing of my analysis, and so many other technical things. And so to be able to compare gene expression in the US, in India, and in China, I would have to normalize them. And there are several factors that you need to keep in mind when you perform this normalization at different stages of the data science kind of process, right? So the process of dealing with that data. Okay, so about the timing of the program. So the timing of the program right now is going to start on June 14th, as mentioned right here. You can see the detailed schedule also on this link as well. And so I will just once again post this link right here. Let's see. What other questions? So I am a bioinformatics postgraduate and want to start my career as a bioinformatics analyst or scientist. Perfect. So Shobhita, having a bioinformatics background is a perfect opportunity to apply yourself to different types of industry jobs. And that's one of the reasons why we also recommend joining the program with these mentors who have that academic and industry background. And I'm sure that they will mention that in their part of the presentation. What are the skills recruiters are searching to shortlist candidates for hiring? Again, perfect question. I recommend also looking at the LinkedIn group that we introduced today. That's where we post a lot of these opportunities for industry internships, research fellowships, and also jobs. And so there you will see some of those questions answered as well. For those that are from a botany background, Saida, I think that's a perfect background because you're already versed in biological data. You know what the data coming from different types of experiments looks like. Now you need to understand how it differs when you talk about biomedical applications and what are the elements of data science that you need to master so that you can apply yourself effectively in these new methods and applications. Will this course cover the interpretation and analysis of multi-omics data? So for that reason, let me briefly show you some of the things that we will cover. And these are related to data processing, analysis, and integration. So what we specialize in is the analysis of omics data and the logic behind that analysis. So how do you actually take a look at a specific data type and understand the logic of how it is prepared and what are some of the questions that you can ask about it. Now, in the context of this platform that I'm showing on my screen, which is what we will use for some of the sessions in the beginning of the program, you will see here multi-omics integration. And so for multi-omics integration, we will take a look at several different methods that are based on either unsupervised or supervised integration so that we can understand relationship between features coming from different omics data types and their relationship. So we will cover that in one of the sessions. It will not be necessarily the exclusive focus of the program, but we will definitely look at that as well. Okay, so thank you everybody for the questions. I hope that I was able to answer some of those questions, maybe not all of them. If you are interested to ask me more questions about this or to ask our team about different programs, please stay until the end of the webinar where many of these questions will be answered. And you can also submit your question right here in the chat box. And later after the webinar, our team will be able to answer those as well. So now let me pass it on to the next speaker. I'll briefly introduce him here, Dr. Sujit. So Dr. Sujit Tangadipiliwar, sorry if I mispronounced your name, is a founder of Pharmatoppers and co-founder of an IT company called Octam PBT. And currently he's a lead data scientist on computer vision projects at Era Matrix. He served in multiple positions in many companies before, like Persistent Systems, Accelera, Hexaware, and others, and played a crucial role in applying artificial intelligence in the drug discovery field. He obtained his PhD in pharmacoinformatics from National Institute of Pharmaceutical Education and Research in Mohali, and he published several research papers in peer-reviewed journals and developed industry-usable softwares in the field of chemoinformatics and bioinformatics in his industry and academic tenure. He has been using his programming skills to solve biological and chemical problems for a long time, and now he's joining this program as a mentor, and he will be taking some of the topics, including the applications of data science in pharmaceutical and biotechnology companies, machine learning and deep learning, natural language processing, as well as chemoinformatics. So let me pass it on to Dr. Sujit.
Thank you, Ilya, for the nice introduction, and thank you for explaining about the project and all about the program, everything about. Thank you. Sonalika, could you please make me host so that I can share my screen?
Yes, sure.
Thank you, Sonalika. Hope my screen is visible to everyone.
Yes, sir, it's visible.
Thank you. Yeah, as Ilya Brodsky said, this is Dr. Sujit Tondon. I'm a founder of Pharmatopus and co-founder of Octane Private Limited. I did my PhD from NIPER in the area of Bioinformatics and drug discovery, wherein I applied several machine learning and deep learning technologies to solve chemical and biological problems during my PhD time. So, being a part of this course as an industry expert, I'll mostly deals with the deep learning, machine learning, all those algorithms, the math, the mathematics behind those algorithms, natural language processing. I'll explain you about what exactly the natural language processing is. This might be a very new terminology for many of you. Computer vision, it is all based on the image analysis, image processing, and all those stuff. And the AI, you might have seen, you might have heard about it. It's a AI, artificial intelligence, is actually a brain of any robot or any intelligence system. So mostly deep learning, natural language processing, machine learning, computer vision is all actually a part of AI or a data science. So into this course, initially we have on a 14th of June, that could be a first session wherein we will deal with the various machine learning algorithm, what exactly machine learning is, what are those algorithms for a machine learning, and a little bit math behind those algorithms. Instead of only learning a theoretical part of a machine learning, we'll also go deeper, somewhat deeper for the mathematics which is involved in machine learning, as well as in a deep learning. So NLP is actually stands for a natural language processing. Natural language processing actually deals with a data called textual data mostly. So let's say you have a set of genes, you have a smile structures in a structure of a molecule in a smiles format, or if you have protein sequences, everything can be dealt with natural language processing technology called NLP. So I'll help you out how we can process those data using NLP technology. As of now, very few industries in India or abroad, they are using NLP technologies for gene or protein data. But yes, we in this course will explore that part as well. On a computer vision side, we will deal with the several images of a drug molecules, wherein based on images of a drug molecule, we could able to find out the toxicity or a biological property of a molecule. So in this course, I'll highlight and I'll share the code which can help you guys to work on a image data to find out the many several biological properties of a molecule. On the 16th of June, we will go somewhat deeper at the mathematics behind those neural networks. When I say deep learning is actually they are a neural networks. If any neural network contains more than one or more than two hidden layers, they are called actually a deep neural networks. So we will go behind, we will go somewhat mathematics, how to tweak those networks, how we can, what are those input parameters, how we can define the output parameters, how we can build a network. We'll go everything about it. And also we'll go somewhat mathematics behind the natural language processing. What are the word corpuses, how we can use word embeddings. I mean, how we can convert those words into a numbers. As you know, computer can only understand the numbers. The computer doesn't know whether it is a protein or a drug molecule or a gene or a or any DNA sequence. Computer doesn't understand, but computer only understand the language of the numbers or a bit 0 and 1. So in this course, we will help you out and we will teach you, will train you on how we can convert those text, I mean the smiles notation or a protein sequence or a gene sequence into the numbers, and how we can process those numbers using natural language processing. In this NLP part, we will also learn some little bit about what are word embeddings, TF-IDF vectorizer, and count vectorizers. Where these are the method to convert text into a numbers. And another algorithm called convolutional neural network. In this is called a CNN. In this algorithm, we will walk you through how we can build a CNN model, what are the parameters involved in CNN model, how we can read those image data, and how we can pull out features from those images, and how we can use those features for CNN model build development, and how we can use those model for the further prediction of either of any of the biological property of a molecule or a protein. in a next session, we will share some of the codes with you. This session could be a completely of a practical session wherein we'll share some Python program. By the time you will learn most of the, you'll get an hands-on a Python and R as well. And also we will share some Python programs with you, and we'll ask you to write some Python program to let's say to calculate the biological properties of a molecule, or to find out the toxicity of a molecule or a protein. So we will also we will help you to write a code for several other biological properties of a molecule, and we have several case studies with us. So, and also we will share those case studies with you, and also will help you to solve some minor bugs or a problem. Those are if you get some bugs associated with that code. So these are the 3 live sessions which are on 12th, 16th, and 14th, 16th, and 18th of June. For other session, I would like Mohit to talk on about the machine learning in data science. Machine learning in biomedical data science. Mohit, over to you.
Thank you, Sujit, for the wonderful overview. Let me share my screen. Hello, everyone. Am I audible, Sadika?
Yes, Dr. Mohit, you're audible.
Okay, thank you. Hello, everyone. My name is Mohit Mazumdar, and I'm one of the co-mentor and project guide for the students or cohorts who are interested in working on their own biomedical project. So about this program and the sessions, as you have already heard from Dr. Sujit, let me explain the next sections of the program. So the purpose of this is to understand about biomedical data science. So data science has the potential, as you know, has the potential to be impactful in pharma and other industries, such as application of data science in clinical trials, and also it helps in cutting down the cost and all of that. So in this entire program, we will be learning about the roles of machine learning and to interpret and analyze biomedical data. So the sessions, as you have already gone through the first 3 sessions, next section will be taken up by Dr. Harpreet Kaur, who is an expert in machine learning techniques and has several publications applying data science techniques in different types of omics data. So in this program, you will be learning about how to load, explore, visualize, analyze biomedical data using big data analytical tools and coding in R and Python. So the session after that, after the first 3 sessions, so this will be the 4th session, and here you will learn about the NGS data types, some of the examples of continuous data, which is the gene expression data, and then examples of categorical data, which is like the clinical and the drug response data. So in this session, you will be introduced to complex data sets, and then we will be helping you understand how to prepare and analyze the data set correctly, and to do some exploratory statistical analysis. So in the next session, which will be the 5th session on 23rd June, we will use supervised machine learning algorithms such as principal component analysis, hierarchical and k-mean clustering to find groups and then to find patterns and similarity in large data sets. In the next session, which will be the 6th session, after learning about the unsupervised machine learning methods, we will be in this session, we will be learning about training and test data sets and how we can apply classification algorithms such as decision tree, random forest, support vector machine. After this session, once we have had the introduction and understanding about different techniques and learn to apply different algorithms using the TBioinfo platform, in the next session, which will be the 7th session, we will be discussing and we will be guiding participants to develop independent biomedical data science project. And as I was telling you about this, that in this session, we will be understanding about the different technologies that were applied in a project and how to do the data integration when we are considering multiple data sets. So after learning about this part, the sessions starting from 30th June till 7th July will focus on coding in Python and R and performing different machine learning algorithms on project or example data sets that we will be sharing with all the participants using our Slack channels or emails and with all the other resources. So in addition to the sessions, the participants will be able to learn by completing the associated tutorials on the Omixlogic portal that you saw. So in the next session, we will be performing clustering on different biomedical data sets and understand what it means in the context of a research. So after this, in this session on 5th July, we will learn about classification and understanding the effectiveness and to differentiate the results that you obtain from different machine learning algorithms. So after this, we will dive deep into the model accuracy and validation, and use for this, we will use examples of clinical and NGS data. And in the 12th session, which is the penultimate session, scheduled session, we will discuss the real world applications of machine learning. And we will also go through the understanding of how artificial intelligence can make a difference in decision making. So in the last scheduled session, we will review the project proposals and we will provide feedback from industry experts on the project proposals and what would be the next steps. So this session will be about planning your bioinformatics analysis, literature review, compiling a primary data set, developing a research plan and methodology, performing exploratory analysis, data processing and preparation, statistical analysis, biological interpretation and validation. I know it sounds a lot, but these are the steps that you need to go through when you go through or design a biomedical project, which would be one of the objectives of this program. And this is what we have prepared for you to go through the biomedical data science program. But along with this program, as a part of this entire initiative, the summer initiative, we will be also doing the Bioinformatics program, which will be going along parallelly. So before we talk more about how you can register for the data science program, I'll also tell you a little bit about the Bioinformatics program, and then I'll pass it on to Ms. Sonalika to tell you more about how you can register for either one of the program or for both the program if you are also interested in Bioinformatics and the data science involved in that chemical space. So the program, which will run subsequently, and like it will be running alongside the data science program. So the sessions are scheduled on different dates. So the sessions does not align. So the students who are opting for both the programs can take it as well. So this program is designed to address the challenges associated with understanding, modeling, screening, and applying chemoinformatics strategies to improve drug discovery results. And this program will be starting on 15th June, and it will be available, and the sessions will be scheduled till 22nd July 2021. So in this program, we also have a 2-day complete workshop. This is all-day workshop. So in this workshop, participants will get practical experience and in-person guidance to use the tools that are offered by Schrodinger. So you will learn about the Maestro GUI and how to do organic molecule sketching. That will be very useful in your own designing the molecule project if you are interested. And also like screening for hit identification and finding therapeutic targets and all of that. It will be available as session 3rd and session 4th. It's planned for the weekend. So we will have several sessions where we will be going through practical hands-on in this one. So you'll learn about docking, then you'll learn about hit to lead modification using software. And then there's a session on free energy perturbation using the molecular dynamic simulation data that will be covered in the 2nd day of the workshop. So these are some of the opportunities that are upcoming. So in all, we are trying to help you develop understanding about different types of data sets and how data science plays a role in it. So there is an important sessions that will be available to those who join the programs that we are offering right now as a part of summer programs. So data science and its application in clinical trials and clinical research. So you'll be getting all of that and learning all application of AI in healthcare using the resources that will be available for you in June, and which will with the entire training will be available for 1 month, and also the access to Schrodinger software license, and also access to the TBioinfo server license. So very interesting aspects for a researcher to go through these tools and to develop projects and show this to in our upcoming symposium in August and get an opportunity to win prizes and also to participate and get feedback from international panel of experts. So all of that is coming up, and for you to start that journey, now I'll just pass it on to Sonalika to talk about and get you registered. Thank you all. And if there are any questions, I'll be looking at it and I'll be answering them in the chat or throughout the session. Thank you.
Thank you so much, Dr. Mohit. So let me take you through the registration process. So first of all, we have this pre-registration page over here. I would be sharing the links with you here. So just to learn about the program more in detail, so we had 4 phases. We are done with 2 of them already. That was on genomics and transcriptomics. And now we are starting with the 4th phase that is on machine learning. And then we have chemoinformatics as well. So to know more about the program and to pre-register yourself, you can refer this link over here. And this is the pre-registration form. You can fill it out. We'll get your entries and then we'll get back to you once we receive your pre-registration form. To register yourself directly, you can simply go to the machine learning for biomedical drug discovery page over here and start your application. I'll just share the page link. So just start with the application and you will get to know the different levels we have for this program. These levels are different based on the provisions you would get, the access and the mentorship guidance you would get. And once you get started with the application, you can simply choose the option you would like to proceed with, select the option and proceed further. And also many a times people want to know more detail of the session. So Ilya has already told you that you could get the session details over here as well, on which day, what topics would be covered. And to get the details on the chemoinformatics program, I'm again sharing this link here with you. So I had a common doubt. People were asking me, what is the registration link for chemoinformatics? So this is the cheminformatics page, and then you have the machine learning page. But for all the programs, our pre-registration form does the same, right? So you can simply go ahead and proceed your application from these pages. Now, if you have any doubts, please let me know in the comment section itself so we can take it here. And to know more about the cheminformatics program, we would actually be conducting a webinar on 3rd June to talk more about cheminformatics in detail and to discuss each and every session topics which would be covered in the program over there. So I'll be sharing the link of that particular session as well with you. And you guys also can get yourself registered for both the programs at just one time by paying for both of them at one single time. And those who have not yet followed us on the social can do so by following us on the LinkedIn page over here, Omixlogic. Now I'm asking you this just because I want you all to stay updated with all the sessions and webinars we have so that you don't miss out the chance of your research interest training. And then we have our Facebook page, though I've shared the link, but I'll be sharing it once again. registration can be paid by invoice. Yes, Anurag, it can be paid by invoice as well. You can mail me at marketing@pine.bio for any of the queries you have, which you're not able to ask right now, maybe. And also, if you face any issues at any particular step, you can mail me and I would be there to help you out. And if you want me to generate an invoice for you, please mail me that as well. You'll get the invoice then and there. And also, we were streaming live for this session we had today. So the recording is here. If any one of you missed out on the important point, you can refer the video here. And also, I would be sharing the recording with you as well. If you guys have any doubt, please ask in the comment section, or else we can end the session. Yes, I would be sharing the workshop recordings as well after each and every session, and those recordings would be downloadable. And during this, so being a part of the training program would get you the access of our portal. You would be able to go through the training properly. You would get the access of a TBioinfo server as well. That would be for the whole length of the program, that is 1 month. And you would also get the mentorship. So during the program, whatever doubts you face, your mentor would be there to help you out. Okay. Okay, so Chitra, I'll send in the group itself. So Chitra, are you already a research fellow or are you a member of the PINE itself? Which group are you talking about? Daniel, you can refer to the page to have a look at the cost of the registration maybe. And we have different levels based on the provisions. Okay, so Chitra. So those people who are there in the Slack channel already, I would be sharing all these links over there and then you can proceed further. Again, I'm sharing with you my email ID. If you have any doubts, please mail me at marketing@pine.bio, or you can also mail Dr. Mohit at mohit@pine.bio. I hope all your questions were answered, and if not, you can mail me and I'll be there to assist you. Thank you all for being such a supportive audience. And if you have any doubts, let me know. Ma'am, data science training program is free. So Chitra, I'll be getting back to you this in the Slack channel itself because you are our member already. So we can talk about this later. Okay, so actually we have the members who are already a part of the Pine Biotech, who are already paying for the research fellowship program. We have a different sort of cost thing for them. So I would be getting back to it later. Thank you so much. Have a great day.